Drug developers, investors, researchers, and corporate executives wrestle weekly to understand what is happening in commercial development of NASH medications. Join hepatology researcher and key opinion leader Stephen Harrison, C-suite veteran Peter Traber, and forecasting and pricing guru Roger Green as they discuss the issues affecting the evolving NASH market from their own unique perspectives on this week's edition of Surfing the NASH Tsunami.
For everyone with an interest in NASH, or more broadly, fatty liver disease, surf's up, because episode 22, day 2 of Digital ILC, is now live and on the air. First, I want to thank everybody who's already listened to day 1. In 12 hours, it had more listeners than any previous episode it had in 24. And in 24, it looks like it's going to have more than anybody had in a week. I also want to thank people who went crawling back through the old episodes because you were so excited. Stephen's 2 favorites, the one about recruiting for COVID in a crisis and what the Intercept complete response letter at the FDA meant, both got new listeners today, which means people liked what they heard and went back and listened for more. There's a lot of good stuff in the old episodes. Please feel free to go do that. Okay. Also, we're doing something this afternoon that we've not done on our previous 21 episodes, which is that we have a sponsored segment at the end. We call it an extra episode. It's about 25 minutes. We'll talk about that later, but today's episode is sponsored by Sonic Insights Medical Corporation, whose device, Velicor, has recently gotten its 510K from the FDA. We'll be talking about that later with the CEO of Sonic and with the person who, the doctor physician who oversaw the poster study that they're presenting here. Okay. With that, I think we can get going. Yesterday we ran long. Today we're going to try to be a little more efficient on time while not losing any content. So here's where I want to start. I thought Donna said something on the way out of yesterday's session that I commented on at the time and will again, which she talked about. One of the nice things about being trained as a lawyer is that you wait for all the evidence before you decide how to judge the story. So we now have more evidence today than we had yesterday because we've been through one more day of sessions. And what I'd like to ask each of you on kickoff is one thing you heard today that might change how you look at the story of what we're learning overall about fatty liver disease at this meeting.
Yes, Louise, I'll be brave today and go first. I listened to the excellent session on case finding in liver disease, and there was a very good GP, Dr. Rachel Pryke, and what she said was that GPs see what they're commissioned or incentivized to see. And in one respect, that saddens me because we need a fairer system where everybody's incentivized to look for diseases. But I thought it was a very honest statement and something that I suppose we've long been aware of, but it was actually really brave of a GP to actually state it. And I think for liver disease in the future, I think we need to be, do we need to be incentivizing for one of the most populous diseases that's affecting the globe at the moment? It's a sad thing if we do.
I think that's an excellent point, Louise. And before we go on, I'm going to introduce, which I did not do initially, today's guest, Vlad Ratziu. Vlad, welcome.
Thank you, Roger.
So do me a favor, take a minute and tell everybody a little bit about yourself and you know what you do in the community and your role, and then tell us one thing you've heard today that has led you to change a little bit of how you think about the story of fatty liver disease.
So I'm delighted to be amongst you here today, and thank you very much for the invitation. I'm a physician specialized in liver disease, working at a large tertiary care center in Paris, and I've devoted the past 20 years or so in trying to set up a research program on fatty liver disease, and along with Stephen and other people, we've been following this field for many years now, with all the difficulties you all know back 15, 20 years ago until now, when we're living through very exciting times, in particular this liver meeting, which would be a fantastic meeting had it been physical and not digital, but still there's a lot of things are presented here. And what I liked today, so of course you don't approach this type of digital meetings the same way and with the same trepidation as you do when you meet your colleagues in the room and can have all the time you want to ask questions and so on. But still, there are a lot of things that were shown in these 2 days so far. And one of the things today that was particularly exciting was the whole session on the new drugs that are in development for NASH. So that is one of the things I'm taking away. And the other thing was we're starting to have a lot of data trying to show that non-invasive markers can somehow predict histological or track along with histological changes on one thing. And the other thing is that now we have some imaging studies, and this was presented today, especially by measuring liver fat content in the liver quantitatively with MRI-based methods that kind of predict a response that goes beyond defattening the liver, and in particular, a response in terms of inflammation and fibrosis.
Okay, thank you, Vlad. Vlad's being a little modest, saying he's been toiling these fields for 20 years. I would argue that in part because of your excellent work and in part because of your low-key personality, I would say you're one of the best known folks in every liver room you walk into. You and Stephen and Donna and the rest of us. Well, I'm kind of a slacker, but that's all right. Who wants to go next?
Hey, this is Stephen. I'll go ahead. So first of all, Vlad, thank you for joining us today. It's an honor to have you a part of this podcast. And I'll just echo what Roger said. I remember you and I, meeting each other maybe 2 decades ago in the early days when I had hair and you were showing me how to do things. So anyway, we've come a long way for the field, and maybe transitioning over to what I saw today in reference to Donna's point yesterday. I feel like we're almost in a tale of two cities, or the perspective I got from today was the General Session 8 abstract that was presented on reflex AST testing in a large population in Wales, just highlighting the fact that a significant number of primary care providers in that region, and I can attest that is in the US as well, aren't really working up patients with elevated ALT that have fatty liver. It turns out in that trial that just looking at a very simple AST-ALT ratio, which is called the DeRiddish ratio, it was originally presented in 1950. I mean, this is something 70 years old. They were able to find a significant number of cirrhotic patients. In fact, of the 2,117 AST-ALT ratios greater than 1, subsequent to that, 348 went on to have a FibroScan, and there was an 81% increase in the diagnosis of cirrhosis in that region of Wales, where they reflexed to an AST and then chased it with a FibroScan. And so while Vlad is absolutely right, we're making huge strides on the pharmacotherapy front. I think every meeting we raise the bar a little bit higher on, the 2-point improvement in the NAS, NASH resolution, fibrosis improvement. Now we have drugs talking about 2-point improvement in fibrosis stage. So I think we're making huge incremental advances there, but at the same token, we still have a huge issue with disease awareness and getting at primary care providers, endocrine GI guys, and giving them the tools and the education that they need to go out and find these patients. So that's my thoughts for today so far.
So Stephen, from a completely different vantage point, or somewhat different vantage point, I wound up in exactly the same place. I was pretty well blown away by that talk and by how much is going undiagnosed and unrecognized. Forecaster, I immediately convert that into market potential, but I also convert that into human loss and sickness. And when you put that together yesterday with, I forget whose paper it was, where at the end they said that abdominal girth tended to be a pretty good proxy that you could use even if you couldn't get tests done to figure out who was likely to be at risk. This shouldn't be that hard to do. We just haven't made it important enough for primary care and for associated professionals to do it.
Well, Roger, I agree with that. The other thing I would say that I forgot I wanted to mention, but I didn't, and getting at this idea that in a way we're behind the times. There was a poster by Michelle Lay from Beth Israel Deaconess in Boston, poster 107, that looked at a large database of patients to see who with NASH were on a GLP-1 or an SGLT2 inhibitor. And I don't have the poster in front of me, but the numbers were surprisingly low relative to what I see in my practice and what I see in my clinical research unit. In fact, it seems like about 15 to 20 to 25% of our patients coming in with diabetes are on a GLP-1. And so while I understand that the data that was presented represents older data, because it's mined data from these large registries, there's been a huge shift in our diabetic management. And as really a second line agent behind metformin, you're beginning to see these GLP-1 agonists come on board. Despite that, we're seeing lots of NASH. So I think that may hold something down the road when we begin to look at GLP-1 agonists in the treatment of NASH, that while it has a role, it may not be the panacea that a lot of people think it might be, but we'll have more time to talk about that.
Yeah, we will. Okay.
So
that
Roger,
leaves, go ahead, Donna.
Yeah. So to Louise's point, incentives do matter. And we do need to build in not only the awareness, but the education, the quality measures, so that physicians have the time and the materials to be able to prioritize liver disease and liver health in a way that they haven't been able to before. And Vlad, you said something in your presentations yesterday about NITs and people needing to believe it to be able to use it and to be convinced of it. And so those two things together, that people need to be incentivized to do it, they need to believe that it has value. And so luckily there's a third day, so maybe it'll provide the third piece of the puzzle. What I wanted to talk about today was specifically about, I saw the persistence of use of the term MAFLD with an M by our European colleagues. And, despite the, fantastically articulated, paper in hepatology with Arun Sanyal and Zubair Younasi and others, which called out why so many of us feel that it's a premature change in term and perhaps not altogether accurate, because there are other types of metabolic diseases that wouldn't be covered because of things that we've been putting into place in terms of awareness or drug development that would be disruptive. I know that there's a core there because of what we've just started to discuss, that there is an element of opportunity to have NASH and fatty liver disease used as a point of entry to make sure that everybody's hypertension and high cholesterol and diabetes are also diagnosed and well treated. But that does not lead to the name change, this choice at this time, without collaboration and involvement of the full field, including the patient community, as we search perhaps for something better as the information continues to evolve, particularly the genetic information. So that's what I noticed today.
Yeah, I think that one of the things I noticed, to move into a little bit of a different area, is that this year we're seeing clinical trials that have drugs with particular mechanisms of action, and those mechanisms of action are proving out in the clinical trials, including things that might be considered adverse, like lipids changing and so forth. And before we were talking about mechanism of action in a very abstract way, not really understanding the underlying pathophysiology. And now we're seeing, I think, a much better correlation between pathophysiology, mechanism of action, and the results of clinical trials. And I'm going to present, so I won't talk too much more about that, but I'm going to present on the fatty acid synthase inhibitor, which I think is an example of that. So I'll be talking about that later. But that's one of the observations I've made, that is different this year than in past years, in my mind.
Okay, thanks, Peter. I'm going to add one more subpoint to the point I made earlier, which is as I was sitting there today, I saw both sides. As I saw what Vlad was talking about, the exciting medications, and I saw what we've been talking about in terms of all that isn't getting done. And it struck me that if big pharma, not the folks who are developing the drugs now, but big pharma were a little bit less gun shy, they would start thinking now about how to prep primary care to think more broadly about the role of the liver in all these processes. And they probably wouldn't have to invest a ton to start to see a lot of difference because there's so much ground that's left to be covered. I don't see anybody doing it. It's not how they operate. It's the world I want, not the world we have. But it would be great if we could start thinking about how to get some private sector money that will benefit in the long run from treating liver more aggressively and more intelligently into thinking about how to build that process now. That was my other thought for the day. All right. So with that said, the agenda for today beyond this is mostly what it was yesterday. Each of us has a paper, will be a presentation we'll be talking about. Take maybe 5 minutes to talk about it, a few minutes for questions, and then we'll see what we've got left for time at the end. Louise, I believe on the randomly generated list today, you were first. So why don't you please go ahead?
Yeah, I was first, and I'm doing AS064, which was actually under an embargo until first thing this morning, so I really didn't get a lot of time to look at it rather than listen to the session, which was great. So it's incorporating assessment of liver fibrosis into routine diabetic review, a primary care pilot. And it was Dina Masur and the team at the Queen Elizabeth Hospital, Gateshead. It was a Berwick Road surgery and Teams medical practice. So hands up to that lot for putting this together, because I thought it was a good session. So basically the overview was what they wanted to do was put in and develop a pathway through routine care when they did their diabetic review. So they were looking at the NICE guidelines, which we've touched on as we introduced the episode, is the fact that it's based directly on abnormal liver function tests, and which the Wales project showed we're not really doing that much, so we're not going to pick up those patients. So implementing the guidelines is a challenge. So we avoid finding a large number of patients at risk, limited time for the general practitioners to do that, low diagnostic yield from the abnormal liver function test. They don't have, or it's very limited access to FibroScan or ELF. So they were looking at a risk advanced fibrosis and complications are increased in type 2 diabetes, particularly those who do use alcohol. And the routine review for type 2 diabetics is about yearly. So they were looking at the screening using sample biomarkers. So the aim was to develop a pathway incorporating a 2-stage assessment of the liver fibrosis on that annual review. So they took 2 GP practices, as I said, 12,175 patients. All of the patients had type 2 diabetes, and they underwent review between April 2018 and September 2019. Primary outcome, number of patients identified with advanced liver disease after the specialist review. Now, unlike the Camden and Islington pathway, what they did was a FIB4 score that was requested through electronic patient records. They used an age-related cutoff for that, either above or below. The GP was then advised to request a FibroScan if it was above the age-related cutoff, and they used the 1.3 for less than 65 and 2 on FIB4 for over 65. So if your liver stiffness was greater than 8, you went through to liver screening, ultrasound, reviewing the liver clinic and biopsy. And if it was less than 8, you had lifestyle advice, management, cardiovascular risk factors, and primary care with a repeat FibroScan in 3 years' time. So the analysis and data of that, the average age was 63, BMI was 32. Missing data, they had a few in the alcohol, but it was generally generic. So if we looked at the results, 477 type 2 diabetic patients attended for the review. of which 467 had a FIB score requested. 18% of that age cutoff went through to FibroScan, 58 were referred for the FibroScan itself. 43.1% of those patients had a FibroScan of greater than 8 kilopascals. 70% had a higher than 8 to 9, and greater than 15 was 22%. So 19 patients were diagnosed with advanced liver disease. One went through to biopsy and was F4. 20% had advanced liver disease. But what was concerning about that was they picked up patients who had hepatocellular carcinoma, or they had large varices. So there was a pocket of undiagnosed, quite severe disease in those sitting in those practices with type 2 diabetes being seen. So that was a concern. ALT wasn't specifically specific, and they found that there was a higher indication on gamma GT for sensitivity of 86% in comparison to ALT at 54.5%. Predictors of advanced disease were age, alcohol intake, and BMI, and gender male. Interesting thing about the alcohol, it was 14 units had a 31% increase of risk against 4.4 at 21 units. So people drinking slightly less were actually at bigger risk. So the conclusions of that study, that the pathway incorporates two-stage liver fibrosis assessment to the routine diabetic review of primary care. It's systematic, standardized, and identified diabetic patients at advanced risk. Ultimately, it showed that there was a 7-fold increase in the detection of advanced liver disease compared to the standard care. 46% had a normal ALT and would have been missed if we use national guidelines. 20% had complications, HCC and large varices, as I said. Alcohol use, increased BMI, male were predictors. FIB4 effectively freer to detect FibroScan in the community was highly cost effective, and that the clinical commissioning groups, of the people who pay for us, develop a business case and roll out this pathway across exploring incorporating reflex testing for serum biomarkers. So for me, that was a great one. I think in relation to what did I learn about the disease, not a lot in that context, but it had a massive impact and the potential for NHS and Public Health England to look at cost. And the more we're finding that cost screening in these patients is highly effective. But when you've got a 7-fold increase in diagnosing diabetes with serious levels of liver disease, we do have a significant problem. So that was the paper that I had the pleasure of listening to this morning.
Louise, this is Stephen. Thanks for sharing that. I'm just juxtaposing that paper against abstract number 8 from Wales. And then I'm thinking back to a paper that Mazen Nareedin just published in Gastroenterology, using a modeling technique to show that it's actually cost effective to screen diabetics for liver disease, which has implications for our practice guidance document potentially that might be incorporated in the next round of guidance. I don't want to speak for the group, but it's something that I think I'm going to advocate for. In reference to the UK, having never practiced there, despite being on staff at Oxford, it strikes me that using simple tools like FIB4 or even AST-ALT ratio, how many people have been picked up, but they're being picked up with advanced liver disease, like at the time when they're presenting with cirrhosis. So ideally, we want to grab these people earlier. My thought is with FIB4, that basically is driven by AST and ALT. So I guess I want to hear your thoughts on whether it would be prudent to first look at ALT-AST ratio. If that is not elevated, then do the FIB4. If that's abnormal, then go to FibroScan, so that we're not in silos here. We can actually incorporate all the learnings from what we're hearing at this meeting to maybe discover patients quicker in that region and probably around the world as well, without having to get very complicated with proprietary scoring systems and various different algorithms. Let's just look at what we get every day: ALT, AST, throw in a FIB4, and then maybe a FibroScan. If utilization, that FibroScan has to be parsed out.
I think you're right there, and I think one of the problems in the UK is we do not get ALT and AST as normal screens in GP practices. So a lot of practices don't have access to the results that they need to make these calculations and to do FIB4 or ELF. So, and when you do ask them, it's a case of it's not committed. I can't afford it out of my budget. Who's going to pay that extra pennies for that blood test? But I think we pay in the long run by poor diagnosis. But these people, particularly the two who had HCC and the two that had advanced varices, by doing this screening and this pilot, it saved their lives and it gave them longer life. And I think that's the key, and it's consistent coming out of EASL at the moment, is adding FibroScan and better non-invasive technology to GPs is key.
And the presenter said the AST was what, 50 pence or something. I don't know how much that is. But
yeah, it's practically not a lot.
Yeah.
There was in one of the industry symposia later, there was, I believe it was Quentin Alsty presenting, but I was hopscotching on different tracks by that point. But I was sent a picture by one of my team. We have 4 people across the meeting to make sure that we get as much of the content as possible, and particularly are seeing the connections between things. But I'm looking at a picture of a really fantastic risk stratification to guide staging and disease management slide that was put together that does exactly this, that looks at the parameters: age, AST, ALT, platelet count, BMI, albumin, impaired fasting glucose for those with type 2 diabetes, and then has a really clear, even color-coded pathway of how to then take the next step with people and what the cutoff points. And of course, we're trying to reconcile what that indeterminate group looks like. But it's really condensed nicely with labs that most people have. So Elise, I think you've just given me my next sort of coding or policy battle, now that we've got the ICD-10 codes fixed in the US, to make sure that physicians do have these available to do some really simple things. When I talk to the primary care physicians, this is the type of thing that they really want.
But I wonder whether or not, in some respects, we do slightly shoot ourselves in the foot, because we use each of these research projects in one disease area, whereby if you combine all of the data that's coming out of using non-invasive techniques as a whole for a GP, actually it's not about just putting FibroScan in to screen for NAFLD in diabetes or alcohol in people who drink too much. It's actually combining it. Once you've got access to non-invasive techniques in your practice, you should use it on nearly everybody, because everybody could be at risk.
But Louise, the joke is the best way to eat an elephant is one bite at a time, right? So part of the question becomes, where's the first bite that the most people will take, so you can start down the path that you're talking about?
So if I was diabetic, I'd be insisting on it, that my GP does some testing to help the GP.
Exactly. And that goes back to the point about incentives and being convincing so that they believe in them. I think that these psychological constructs and these cultural constructs of the way medicine is practiced are something that we can't, forget while we're putting the pieces of the science together.
I would like to throw in there a word of caution, Roger, if I may,
Sure.
about the use of FibroScan outside of the hepatology office. In our hands, FibroScan is not always an easy tool to use. There are a lot of people, it's hard to get a reading. There are a lot of failures to the measurements. There are a lot of false positive. You have to know the situations where you cannot have a reliable reading, and the situations where, despite a reliable reading, the result is influenced by other factors, such as an increase in ALT, such as inflammation, such as hepatic congestion, and so on. So it's not that trivial to use FibroScan. And in my view, FibroScan should be something that needs to be reserved somehow to the specialist. You have 2 different probes for doing FibroScan, and then you have other portable ultrasound-based machines that measure liver stiffness that are based on shear wave or supersonic type of imaging methods. And sometimes you have to combine all those to have a reliable assessment. So I don't think there is much place for FibroScan outside a tertiary care specialist office in doing that. I think we have enough things that we can use as a first triage system, such as, look what EASL is recommending, the liver function test. Only that, if you have persistent elevations in ALT, AST, that's enough of a reason to send the patient to a specialist. First, because they can have other things than NASH, of course, other like liver diseases that have to be diagnosed by specialists. And second, because these are the people that anyway need to have an exhaustive assessment of their liver condition. So that's as simple as that. Ultrasound that shows steatosis, liver function test, and then you can throw in there people who have normal ALT and AST, a simple serum fibrosis marker. But FibroScan, I think, is a little bit too complex to interpret in many cases, so that it be used in outside tertiary care practice. The last point I would like to make is that all this, you started the presentation by referring to diabetic people, right? So this whole work was done in diabetics. People tend to forget.
Type 2 diabetics.
Type 2, yeah. People tend to forget that at least half of the patients that we see with NASH that are included in clinical trials, so meaning they're F2, F3, right, with active NASH, half of these don't have diabetes. And Roger, I listened to one of your excellent podcasts some time ago. Actually, it was that podcast where you discussed the CRL that Intercept received, and you had a guest from the FDA. And that guest from the FDA said one thing that truly shocked me. And I know Stephen and Peter were shocked as well. He said, ultimately, if you have NASH, it's a failure in primary care to take care of diabetes. He said that. So,
a former FDA.
He's not an FDA now.
Yeah.
And Donna and I started our next conversation by how shocked I was by that statement. I remember it vividly.
I was not shocked because I've met him, but yeah, he's on my board.
I'm sure many people think like him. So it's just remember, NASH is not only about diabetics. A significant proportion of people with advanced NASH do not have diabetes. So you need a broader net.
Okay, so Vlad, I feel compelled to let Louise respond to the comment, the part of the comment about FibroScan, and then I want to go on because we're going to wind up where we were yesterday and keep talking until 5 o'clock. Louise, please go ahead. 11 o'clock for you, Vlad.
Yeah, no, I absolutely agree with Vlad's summation. FibroScan is a skilled tool and it has to be kept within skilled users, but actually you can put skilled users into primary care to ensure that quality has been maintained. And I think that's what I'm about, that's what I've developed, and it's pathways like this that prove it's highly cost effective. To get a FibroScan in the NHS costs about £700 per patient. That is not something that CCGs can afford. And I know it's placed slightly differently and paid for by Medicare within America, so it's a different situation, but I absolutely fully agree with Vlad. It has to be done by people who are highly skilled to be able to get over and understand all of those quirks.
Okay. We could do that one for a while. We're not going to. We're going to go on to Stephen and a very different conversation. So Stephen, you're up. You're muted.
Okay, here we go. I had the task of presenting or listening to Abstract 77 and talking about it today. This one is hopefully pretty easy for me since this is data I'm pretty familiar with. Ultimately, the title of the abstract was Magnetic Resonance Imaging Proton Density Fat Fraction, MRI-PDFF, to predict treatment response on NASH liver biopsy, a secondary analysis of the resmetirom randomized placebo-controlled phase 2 trial. And Rohit Lumba presented that today. So kudos to Rohit for, while all these are pre-recorded, he was up very early on the West Coast for the Q&A session, and he looked like he had, been up for several hours. So he was rocking and rolling. But ultimately, just to give you a little background, resmetirom is a thyroid hormone receptor beta agonist. It's in phase 3 clinical development for the treatment of NASH with F2 and F3 fibrosis. Currently, there's also a phase 3 trial in looking at milder forms of disease, only using non-invasive testing, well, predominantly using non-invasive testing to identify at-risk fatty liver patients. This data we talk about today comes from the phase 2 paper already published. And in that trial, just to give you a little flavor of the study, if you don't recall it, it was a 36-week trial. It was a paired liver biopsy study. You had to have NASH with a NAFLD activity score of 4 or more with F1 to 3 fibrosis to get in. Patients were randomized 2-to-1 to resmetirom or placebo. The dose of resmetirom was 60 to 80 milligrams. That dose was adjusted based on 2-week exposure of drug or drug level, if you will. So it could have gone up or it could have gone down. 125 patients were enrolled overall. And the primary endpoint was PDFF change at week 12. And just to highlight the primary results, when you look at all comers, 36% relative liver fat reduction with the drug group compared to 14% for placebo. And then more specifically, there appeared to be a dose response relationship between the 60 and 80 milligram dose. There was also a 36-week open label extension trial. So for people that had persistently elevated ALT, whether they were on placebo or on drug, they were rolled over to open label extension, meaning if you're on placebo, you now got drug. If you were on drug, you stayed on drug, but pivoted to a higher dose, 80 or 100 milligram. So we also have 36-week data on PDFF in that group. And there, again, a dose response relationship. So 60 milligram had a 39% relative reduction in liver fat. It was 50% for the 80 milligram, and it was 64% with the 100 milligram dose. But maybe more importantly, what we gleaned out of that initial data set was that resmetirom responders defined by at least a 30% relative reduction in liver fat had higher rates of NASH resolution on the week 36 liver biopsy compared to non-responders, which was 4%. And that caused a lot of questions, a lot of hypothesis generating questions, which led to this current abstract. So ultimately, going back to the phase 2 data, 107 NASH patients, about 73 on resmetirom and 34 on placebo that had paired liver biopsies were evaluated in this analysis. And the take-home point here is that the predictive accuracy of PDFF response for NASH resolution was found to be quite high. In fact, the AROC was 0.89, with the optimal MRI PDFF reduction found to be 41.5%. That was highly significant. So, so just diving into that just a little bit more, Rohit showed a slide in his presentation where he looked at the relationship of PDFF response to biopsy component response. First looked at the NAFLD activity score, and he subdivided patients into those that had higher than a 30% PDFF response and those that had a lower response. And what you see is statistically significant, better improvement in the NAFLD activity score in those people that had a higher than 30% PDFF response. And the same could be said for each of the components, not just steatosis, which is what we all were led to believe, that the change in PDFF just is reflecting a steatosis change. But maybe emphatically, there was highly significant change in ballooning as well as lobular inflammation. And then maybe most surprising, it was also statistically significant for fibrosis improvement. And then finally, the take-home slide I thought that was worth mentioning here is the association of PDFF response with NASH resolution and fibrosis reduction as we move up the response rate from a 30% response rate to 40% to 50%. The so-called super responder, where if you have a relative 50% reduction in liver fat, that we would call it the super responder. So when you look at this figure that he showed on NASH resolution, you see a dose response relationship, if you will, meaning the higher the PDFF response, the higher the NASH resolution response. So that in this graph, if you had more than 50% PDFF reduction, you're actually at higher than 60% NASH resolution, compared to if you just had 30% or higher liver fat content reduction, it was around 37%. And then if you were an MRI PDFF non-responder, meaning less than 30%, you didn't really respond at all. And that held true for fibrosis reduction, and then also for NASH resolution and fibrosis reduction when put together. And I think that's important because that's a higher bar. And we're beginning to see that happen with some therapies in development where we're reporting out both NASH resolution and fibrosis improvement. So I'll stop there and see what we have as far as questions.
Go ahead. Who's got questions? Vlad, go ahead.
Stephen, of course, this is very valuable data. Nonetheless, two questions, simple ones. Don't you think a lot of this is related to the mechanism of action, meaning that works wonders for drugs that are supposed to induce a reduction in the lipid fat content of the liver? So, because once fat goes away or the lipotoxic species go away, and then you have an improvement in fibrosis and so on. Makes a lot of sense. But other compounds that do not primarily act in reducing lipogenesis, in improving insulin resistance per se, other compounds that are purely, I would say, if there any such compounds exist, anti-inflammatory or even anti-fibrotic, you might not be able to use that for measuring this. The second question is, would you really measure the efficacy of a drug by the magnitude of reduction in PDFF? Do you really need, is a drug that has 70% reduction in PDFF at 3 months or 4 months, will it be necessarily a better drug for improving the overall liver histology 2 years later than a drug that only does 30%? Remember OCA, which is for the moment the only drug that has shown improvement in fibrosis, has a very, very minimal reduction actually in the amount of liver fat.
Yeah, those are great questions. And you and I debated this and had this conversation before at various advisory boards. And I think they're both fantastic comments. And just to your point about mechanism of action, I mean, we saw that with seladelpar, for instance, where there really wasn't movement in PDFF, but we at least preliminarily saw some changes in histopathology. Anxious to see that data, hopefully at AASLD. But I do think it's mechanism specific. Obviously, if you're studying a drug like, CRV431, Hepion's pan-cyclophilin inhibitor, it's not purported to have any effect on steatosis, but is billed as an antifibrotic. I wouldn't expect to see PDFF move. Other anti-inflammatory mechanisms, SSAO inhibitors, ASK1 inhibitors, I wouldn't expect to see that happen. In the FXR field, it's interesting. The second generation FXRs, we're beginning to see quite a bit of fat movement. I remember Metacrin having some, actually a poster at this meeting showing some change. You're going to report on Enanta in a few moments, or did already. We'll talk about it today, showing some decent movement in PDFF. So, but to your point, I think it is mechanism specific. But where you do see it move, that's where I'm getting excited about what that could tell us relative to histopathology. And I think the jury's still out on the magnitude of effect change. I think we're pretty convinced that 30% is this magic number. And there may be a threshold at which you don't see additive histology benefit. And maybe that is just specific to a THR beta class where you see this additional benefit above and beyond 30%. But it at least is an exciting tool to help us. And I think just circling back around to the CRL for Intercept, if you could provide the agency, these regulatory authorities, with compelling data in your NDA application that says, look, the totality of the evidence supports the semi-quantitative assessment on collagen and NASH resolution that we found using pathology interpretation. We have all this evidence supporting it. And not only that, but we can use this to help predict outcome or help predict response to therapy. So we know when to stop therapy, when it's not helpful, how long to treat, and that sort of thing. So I think where you're able to show with a specific sponsor, with a specific mechanism, a very specific non-invasive test that corroborates the findings that you're seeing histopathologically, that will be helpful. But it is going to need to be done by each of these mechanisms specifically.
Yeah, I would just add one thing to Vlad, is that, and even presented at this meeting was the nidufexor data, which really showed to me a surprising number of patients that had a greater than 30% reduction in MRI-PDFF. And I think we are seeing some differences from in the non-steroidal FXR agonists that could be very interesting.
Moderator note, we're at 2:45, at 3:15, we've got to be on the extrasode thing, and we've got 4 papers left to do.
I'll be brief.
I'm going to give you a 30-second summary of mine, tell you what I'm thinking. You can comment on it. I'm going to go back and re-record it after everybody else goes, which is what I wound up doing yesterday. But let's see if we can keep the rest of these to 7, 8, 10 minutes. Donnie, actually, you are next, so go ahead.
Less. Yeah.
Go ahead.
Thank you. So I am presenting on AS017, a polygenic risk score for progressive non-alcoholic fatty liver disease risk stratification. It was presented by Cristiana Bianco out of Italy. And some of the reasons that I was interested in this, I am married to a geneticist, and so I guess that has had some influence. I did serve on the board of directors of the Personalized Medicine Coalition around genetic diseases and factors in diagnostics. And I've always been continually searching for examples to feature in liver disease so that we can provide more personalized care for liver patients. So I think this whole line of research and thinking is important. The aim of this study was to come out with a genetic risk score to predict not only NAFLD and NASH risk, but to really tie it to being able to predict hepatocellular carcinoma risk. And so, one of the things I've and my team from the Global Liver Institute are looking for across the meeting are places of these linkages, places where we come out of silo. And so what they were specifically looking at were several different genetic markers, including those that are very well known, like PNPLA3, and combining them into their own sort of proprietary GRS, or genetic risk score. And they adjusted for age, sex, BMI, and presence of diabetes. And they were specifically, she talked through the drivers of hepatic fat accumulation. And so what their results were a score that they believe can, strongly predict liver cancer, even in non-cirrhotics, independent of their obesity and their fibrosis score or stage. And so I think this is to the point of our earlier conversation, where we want to make sure we don't leave those patients who, aren't so easily identified as already having advanced disease. How can we create a way of identifying and, even more importantly, stratifying these patients? And some of the questions that came up, I think, are helpful to how whatever impact this research and the study will have moving forward. There were questions about whether this risk score could be combined with non-invasive technologies that had a negative predictive value to not just include patients in terms of those who might need extra screening, but can you exclude, so that we're devoting resources in the most appropriate places. Another question that was asked, but was answered in the negative, so more research to come, was if they had looked at extrahepatic risk. and that was not a central part of this study. So the conclusion really was that this was, additive to the other types of clinical information that we might have for patients, but I believe it is a very important type of information to have, particularly for patients who don't present with obesity or diabetes, and we don't want to. We want to find an efficient and cost-effective way to make sure we are not missing those patients. And so I thought this paper was very interesting.
So I agree with you. I was powerfully struck by two things about the paper, one of which you've mentioned and the other of which goes to a lot of what we were talking about yesterday. A lot of the cost in the system for dealing with liver disease patients will be the cost of dealing with fast progressors. And if liver is like most diseases, a lot of the fast progressors are going to come out of the places we're talking about, not folks who have the typical pathway, obesity, diabetes, et cetera, but folks who have more uncommon modes of action or modes of disease and are much harder to identify until late in the game, and then it's too late. I saw a guy heading a coalition talking at some Roth event a couple of years ago whose mom had basically gone in 6 weeks from thinking she was fine to being dead and had never been obese in her life. So my thought there was that if we can find those patients, that's a real benefit. The second thought was that if you take the work that they're doing and you add it in, as they said, to non-invasive tests and other forms of artificial intelligence, we should over time become more efficient at pinpointing ways to think about who those patients are. So I just thought it was a really interesting paper with a lot of downstream benefits to it. I was very impressed by it. Other comments?
Yeah, very briefly, if the issue is to identify people at risk of HCC, we need to keep in mind two things. First of all, that even in cirrhotics, the risk of HCC is highly heterogeneous. There are some low cirrhotics at low risk, and cirrhotics at medium risk, and then cirrhotic patients with NASH at high risk. So there might be a place even among cirrhotics to use these genetic scores to better adapt monitoring programs. And of course, the problem is even more important in non-cirrhotics, where we have no clue who develops liver cancer and where these scores could help. The problem is that most of them have a very, very low positive predictive value.
Vlad, I think you're right. And, I think about both of those points, the fact that cirrhotics may progress at different points and that people can progress to liver cancer without having been cirrhotic, which many people don't, not in hepatologists, of course, but many folks, as we're expanding, who we talk to about liver disease, don't know. And so every time I see that linear progression, of advanced degrees from, stage 3, 4 to cirrhosis to cancer, I want to find some way to call out it's not always a linear progression. So thank you really for making that point.
The paper I'm scheduled to talk about today is AS015, therapeutic effect of a novel long-acting GLP-1 GIP glucagon triple agonist HM15211 in animal models of NASH and fibrosis. This is about a compound from Hanmi Pharmaceuticals that's been around for a while, explored in several different things, and they finally landed on NASH as a good use for it. What I didn't realize when I signed on for this is that this was one of 3 presentations on this molecule in the study. This one is theoretical animal models. There's one about their phase 1 safety trial, which is FRI-115. And then there's a late-breaking poster, LBP03, that talks about their phase 2A. And when you look simply at AS015, it's interesting, but when you put it together with the other 2, it becomes a pretty compelling story. So I want to focus on all of that at once. First of all, the molecule itself, HM15211, is a single moiety combining a glucagon agonist, a GIP-1, and a GLP-1. Each plays a unique role in the process of NASH. The GIP and the GLP-1 have impact on hepatic fat reduction and anti-inflammation, in both cases through calorie intake, in both cases through immune cell recruiting and liver macrophage activation. Plus, GLP-1 has an impact on ALT, and the GIP has an impact on ectopic fat accumulation. They believe the most interesting element is the glucagon, which has liver-targeting effects of glucagon, hepatic fat reduction, anti-inflammation effect, bile acid production to decrease liver injury, and then direct fibrosis effects based on GGF-beta and the inhibition of fibrogenesis. A triple molecule with a PEG linker combining the triple coagonist to an glycosylated fragment that gets it to the liver quickly and cleanly. What they set out to do in the AS015 paper was to run separate tests to demonstrate diet-induced effects, chemical-induced effects, and surgery-induced effects that they could create to demonstrate that they had impact on obesity, NASH, liver inflammation, fibrosis, liver necrosis, and mild to severe fibrosis. In the diet model, they fed two groups of 7 rats each, a 40% fat calorie diet, 22% fructose, 2% cholesterol, and then they induced 12 weeks of drug treatment with both a HM15211 cell and a beta-cholic acid 30 mg per kg cell. And what they demonstrated was that in terms of the various fat measures, that obeticholic acid had a significant effect, but that the effect of the HM15211 was more significant. The same was true in blood ALT. And they looked at 2 hepatic inflammation markers. One of them was F480, where the obeticholic acid had no effect, and the HAMI molecule did, and IL-1 beta, where in fact neither had a significant effect. So from there, they also took a look at differences in NAS activity score, NAFLD activity score, where in fact there was 0 for ballooning and 0.1 for steatosis in the HM15211 group, and no really powerful difference, significant but not powerful, in the OCA group. And in terms of NASH resolution, only one of the OCA mice showed resolution, all 7 of the AML mice did. They then demonstrated similarly for in terms of liver damage and fibrosis, they instituted thioacetamide, which is a hepatotoxin, in the rat. They did that for 16 weeks. For the last 8, they had drug treatment. At the end of 8 weeks, there was significant damage. At the end of 16 weeks, the damage had basically been reversed completely. And when you took a look at a serious red positive stain, there was significant reduction there as well. Finally, in terms of surrogacy, they did a 2-week bile duct ligation and drug treatment throughout that, and then they reduced the ligation at the end of 2 weeks. And what they demonstrated was that they could create different levels of fibrosis, but whichever they could return to baseline at the end of the period. So that was interesting in animal terms, but not clear exactly what it might mean for people. It becomes more interesting when you go over to the second poster, the late breaker. In the late breaker, they did a 12-week 1B2A trial looking at 5 different doses of HM15211, 0.01, 0.02, 0.04, 0.06, and 0.08 mg per gig, or placebo in a ratio of 3-to-1, 12 subjects per cohort. They set out to enroll half men and half women. They set out to enroll people with high BMIs and reasonably and liver fat levels on the average of slightly under 20%. And then what they did was they took a look at 8 weeks and then again at 12 weeks to determine what the impact of the different dose levels were. And what they showed was a pronounced dose-related effect. At the highest dose, they literally had 100% of patients demonstrating a 50% reduction in relative liver fat by the 12th week. That number was in the range of 2/3 in the 0.06 group, and then it slid progressively down from there. All that occurred in a trial with highly tolerable gastric side effects, and the effects only occurring maybe 1/3 of the patients or less, which is important because if you look at the history of the GLP-1s in diabetes and the other reported GLP-glucagon combinations, for example, a cotadidide paper reported today as well, there's a significant gastrointestinal side effect that can stretch out for months. So what you see at the end of this is a medicine that has a decent theoretical background in rats and a fairly strong level of execution in its 1b/2a clinical trial. Subsequent to that trial, two things happened that I think are even more compelling proof of concept commercially than simply what's in the late breaker. They were given fast track status for their 2b, and Merck invested in the molecule for space and NASH. So in conclusion, this appears to be an approach that has some value greater than we might have expected just by looking at what's been reported in the GLP-1 glucagon combinations to date. And it combines high level of apparent or seeming early stage efficacy at higher doses with what appear to be a manageable level of side effects. So that's what I have. Questions, anybody?
Roger, I think this was a really very interesting set of papers, and the clinical results are strong. As you said, some of the strongest we've seen. While preliminary in phase 2A, I think the results are very strong. I think this approach of combining different drugs within a single molecule is really quite important. And if you can get activity for GIP, glucagon, and GLP-1 all in the same molecule, and by the way, there are companies trying to even put additional activities into so you could have quadrivalent type of molecules. I think it's a very interesting report, and this is the first time that somebody has actually shown in humans that what you see in animals is extended to these multiple components. Now, there are, of course, GLP-1 glucagon compounds out there, cotafetide, which was presented yesterday. But I really think this is an important trial, and the fact that there weren't overwhelming GI adverse effects makes it quite promising. And then finally, you said something at the end, which I think is really critical. Merck has invested in it. I don't know the structure of that deal. Maybe you can expound on it. But when there's a compound that's been around for a long time and the company is pushing its activities and so forth, and there are no pharma companies interested in it, you have to scratch your head. But with Merck investing in it, I think it could be a very important approach.
Okay, thanks, Peter. Louise, anything you want to add? I do have one more thought, but you have anything you want to add first?
Yeah, the one thing that I thought of looking at the data, and I know it's certainly from the poster, it's a phase 1 study. So although the side effects look mild in the 0.08 milligrams per kilo group, 44% of the patients did get moderate side effects. That was only 9 patients. So if you extrapolate that out, that could be significant when we get a larger volume of patients. And the ethnicity diversity wasn't particularly great, but again, it is only a phase 1A/B study. But if I'm right by looking at the figures, the ALTs were practically normal in all bar 2 of the study groups, the 0.04 milligrams and the 0.08 milligrams per kilo. And we've used that a lot, and we've discussed it a lot across the conference, that's one of the markers that people look for. The fact that we're looking at dynamic fat fraction, I'm just thinking straight off here, how are we going to practically monitor that in the real world if this is as successful as it is? Because not every patient is going to have dynamic fat fraction. But maybe you could both comment on that.
That's a fantastic question. I suspect that, as you point out, it's a 1b/2a, not a lot of work, not large populations. I would guess that as they dig into this, they will find that it makes more sense in some patient populations than others. At that point, I'm sorry, let me start that answer again. Louise, that's a fantastic question. As we will talk about later today in the sponsored section of this discussion, I expect we will see emerging technologies that will purport to find, or will report being able to find, less expensive, more efficient ways to get at things like fat fractions than what we've got right now, which is MRI. So one possibility is that over time, the market adapts to be able to provide what you're asking for. The second is, as they go into larger patient populations, if I were Merck, and no, Peter, I don't know very much about the structure of that deal, one of the things I would be looking to do is to segregate patients so that I can identify groups that were particularly likely to have a good response here. I'd start with obviously high BMI patients, and then I would look to see what else we could add on top of that. And either build PDFF into the protocol for those patients, or we might learn that we could look at GGT or another one of the fractions and get something, or one of the non-invasive tests and get something of greater value, less expensively. I think we're too early to know because we don't know enough about where the tests are going to go. We don't know enough about this product. But I think your point about ALT is an excellent one.
And I suppose in a larger population, the average ALT AST will actually be significantly higher than was shown in this study.
This is preliminary. It's in a small patient population, but clearly suggests going to the next step into a larger trial where they more clearly define their baseline population, and of course have paired biopsies, which is going to be required in a phase 2B. Mm-hmm. So I think it's promising, but it's the verdict is still out for a long time as to whether this translates to a more severe population, as Louise points out, in terms of the ALT, and how it affects other components such as fibrosis. But I do think it is, as I've said, promising and something important to come out of this this meeting.
So the paper that I'm going to review is novel first-in-class fatty acid synthase inhibitor, TVB 2640, versus placebo, demonstrates clinically significant reduction in liver fat by MRI-PDFF in NASH. And it was very nicely presented by Rohit Lumba, who nearly had his own session this morning. But also I want to point out that both of our distinguished panelists here, Vlad and Stephen, are on this presentation. So why is this important? Well, the synthesis of fatty acids, or de novo lipogenesis, fatty acids and triglycerides, really plays a central role in NASH pathogenesis. There's been a focus on reducing de novo lipogenesis and lipotoxicity through a number of different mechanisms. One are the ACC inhibitors, which inhibits acetyl-CoAse a little bit upstream. The fatty acid synthase inhibitors, which is the subject of this one, is just downstream of that. And then there are the DGAT inhibitors, which inhibit triglyceride synthesis. So there's really been a focus in pharma on inhibiting de novo lipogenesis. And to me, this paper demonstrates some very effective reduction. And I want to go through it. The trial is the Fascinate-1 trial. 99 people randomized 1:1 to either placebo or 2 doses of the drug. The high dose of the drug, 50 milligrams, inhibits de novo lipogenesis by 25%. It doesn't completely inhibit it, although it can completely inhibit it. So there's because it's a systemic drug and lipogenesis is involved in a lot of things in multiple tissues, they've inhibited it at 25%. They treated for 12 weeks and they enrolled people with greater than 8% fat with a 15% mean fat. The endpoints were labs and MRI-PDFF. It was generally safe and well tolerated. And the mean relative fat reduction was dose dependent with minus 28% at the high dose of 50 milligrams. The responder frequency was meaning greater than 30% reduction of MRI-PDFF was 61%, with placebo at 11%. So a highly significant difference. There was a dose-dependent reduction in ALT, and 50% of the patients had a greater than or equal to 17 units per liter decrease in ALT, which has been associated with histologic response. There was a decrease in LDL-C and a small decrease in HDL-C as well. So one good, one potentially not so good.
So, Stephen, you had asked me this morning, I guess, one question about about covering this paper. Is there anything you want to ask Peter about it?
Yeah, I think you did a fantastic job of going through the data, Peter. I have to admit, it's kind of interesting to see this mechanism of action work at the low dose as well as it did, and not have some liability associated with it that could be detected. And I'm just thinking back to the ACC inhibitor class, which works a little bit upstream, where we see a significant rise in triglycerides. Do you think we'll see more at the higher dose, or you think this is going to be a clean drug?
Well, that's probably the most interesting part of this, Stephen. And the fact is that, yes, with ACC inhibitors, you get an increase in triglycerides, even though you inhibit de novo lipogenesis. And people are trying to get around that, or companies are trying to get around that by adding other drugs like DGAT inhibitors and so forth to inhibit the increase in triglycerides, which is kind of a counterintuitive way to approach drug development, in my view. But the fact that this FASN inhibitor did not show an increase in triglycerides may be counterintuitive, but it may also be related to the degree with which you inhibit the enzyme. And you saw in the paper that with 150 milligrams per day, they're able to 100% decrease the de novo lipogenesis. Well, what would happen at higher than the doses they use to triglycerides? So to your point, it doesn't alleviate the fact that they may have an effect on triglycerides, but I was both surprised and I think encouraged by the fact that they got such a good response without that adverse effect.
Other
I may add,
questions? Vlad, go ahead. Vlad, go ahead.
Yeah. So this was a very, very good presentation of the data. And of course, it's very exciting. Now, I would add SCD1 inhibitors in the list of lipogenic inhibitors of lipogenesis that are now in the clinic, because we have one of those in phase 3, which is Aramco.
Aramco, that's true.
Right.
Yeah, I wasn't. Yeah, go ahead.
The two questions here are the following. First of all, what's nice about ACC inhibitors, which cannot be used alone because of hypertriglyceridemia, but what's nice about them is that they do have, at least in the animal, anti-fibrotic effects. So I don't know whether this FASN inhibitor has some direct sort of pathway to inhibit fibrogenesis. And I think it would be good that we develop drugs that have this potential for an anti-fibrotic effect. We'll see if it's enough monotherapy or not, but it's good if the drug already has that. The second point is, yes, lipogenesis, de novo lipogenesis is important as a source of hepatic fat, but we all know it's not the most important one quantitatively, which is still lipolysis from the adipose tissue enhanced by the lack of insulin sensitivity of the adipose tissue, the loss of insulin sensitivity. So I wonder, in people who don't, I wonder if that is sufficient truly to improve the liver if you're not correcting for the insulin resistance itself. After all, a lot of these people eat a lot of sugars, but some of them don't, or at least say they don't. And I wonder in this subcategory whether lipogenic inhibitors would be as effective.
Yeah, those are all very good points. And what I would mention is that they did show evidence of improved insulin resistance with an increase in adiponectin, which probably is related to that. Also, FASN is expressed in a lot of tissues in the body, including adipose tissue, and you would expect to inhibit de novo lipogenesis in adipose tissue as well. The other problem is it's in a lot of different tissues which could cause adverse effects. The final thing I would say is that the only evidence they have that it might work in fibrosis is that TIMP1 measurement, which is relatively weak. And I would agree that ACC inhibitors have shown a direct effect on stellate cells and improvement in fibrosis. But we're finding that the lipid pathway is directly involved in stellate cell activation across the board. And that includes not only de novo lipogenesis, but also the ceramide pathway and others. So a lot of these drugs that affect hepatocyte lipid metabolism may also affect stellate cell transformation to myofibroblasts. So, but your point is well taken. The ACC inhibitors are further along in showing an antifibrotic effect.
And I'll just add, wouldn't it be cool if this class of drug did show a histopathologic benefit to support the non-invasive test? It's oral. So thinking about down the road combinations with something like a low dose FXR, THR beta FASN, I mean, there are all kinds of combinations you can think of. It's great to have a different MOA that's oral with a low AE profile that's safe and well tolerated that we could potentially combine. So I think, more to come here.
Yeah, and it is as you guys know, I mean, you were involved in the study, but it is a fairly hot area where people are looking at liver-restricted FASNs and other ways to manipulate this. I think the liver restrictedness is for the reasons that Vlad brought up of adipose tissue is probably not the direction to go. But I thought the paper was encouraging, and I was pleased to be able to review it. So congratulations to both of you guys for being on this presentation.
Well, thank you, Peter. That was great. And Vlad, I guess you're the last guy up today.
All right. So this will be about this class of drugs that we're really waiting with a lot of anticipation to see what they're good for, which are the second generation FXR agonists. So those are non-biliary FXR agonists. So the bar is very high because OCA, the first generation FXR agonist, which is a bile acid analog, has shown, and it's the only drug so far, we need to go back to the essentials, that has shown histological efficacy in a large phase 3 trial. So the bar is really high. Now, the premise here would be to try to develop second generation compounds that, because they're lacking the biliary structure, might not essentially have the enterohepatic cycle. So therefore, they might be devoid of the side effects, particularly pruritus and the lipid deleterious side effects that we know with first generation FXR. And there are many of them in development. And this is one that comes from Inanta, which is a company that you all know that developed very potent DAAs, direct antiviral agents for HCV, and now is into NASH drugs as well. So this is one of the first non-biliary FXR agonists that has been developed by chemists, and subsequent to some phase 1 studies where a range of doses has been established that in the short term at least did not show pruritus and lipid changes. This Argonne study was initiated, and the Argonne study was an international study where 2 doses of the drug were tested against placebo for 12 weeks. It's one of these very nice standardized phase 2A trial designs that we now have, because at least for these metabolic drugs, we now have the non-invasive measurement that allow us to have an indication, first of target engagement, and second, of the physiological effects that these drugs are expected to have. And it's fortunate that, at least for the metabolic drugs, we have this, both the biomarkers in the serum and the imaging methods with MRI-PDFF that allow us to tell whether the drug works, hits the target first and works or not in humans. And so this study had as a main primary outcome ALT reduction, and the results are quite clear. Two doses were compared, 1 and 2.5 milligrams. So people that were included all had a high MRI-PDFF. They all had elevated ALT, since this was the primary outcome. And half of them had histologically documented NASH, and the other half had type 2 diabetes with PDFF above 8%. And this was done in non-cirrhotics. And a lot of people in this trial were from Hispanic origin, Latino and Hispanics. Between 40 and 50% of patients were Hispanics in this trial. And like in all other NASH trials, between a third and half of these patients were on statins, meaning that all the others were not. So the primary endpoint was positive as far as reduction in ALT. There was a very nice dose-related response in ALT, with the highest dose reaching significance versus placebo. Quite higher level of placebo response in terms of ALT, but still the high dose was reducing ALT at a much larger in a much larger degree and fashion than placebo. The second result that was important is on the PDFF. This went along with a strong reduction in hepatic fat content, which is not surprising, as you mentioned earlier, because we now have other second generation PDFF MRI FXR agonists, such as Silofexor, the one that was discussed here, Tropifexor as well, that has demonstrated the same thing. Now, what is interesting too is that whether you measure the effect on PDFF reduction, this is going back to what Stephen discussed earlier, which is the correlation between PDFF reduction and histological improvement in fibrosis and steatohepatitis. Well, whether you measure it by absolute change, 5%, or relative change, 30% from baseline, both cases, clear advantage of the high dose of this compound. Now, a very interesting slide, in my opinion, was the one that tried to see the relative reduction in ALT in the placebo and the active arms, depending on whether MRI-PDFF was significantly reduced or not. So that slide compared people that did not achieve 30% PDFF reduction, and then the different doses and the ALT reduction in those, and then people that did achieve the 30% reduction in PDFF, and then the ALT reduction for those. And what is interesting is that, of course, as anticipated from what was said earlier, the more liver fat defattening you have, the higher the response on ALT. But what is interesting here is that when in responders, PDFF responders, meaning massive defattening of the liver, the amount of reduction in ALT was much higher than that of placebo. So occasionally placebo patients had a 30% reduction in PDFF, but they did not have a massive reduction in ALT that went along with it. And this is an interesting result because it tells you that you need PDFF reduction to reduce ALT, but probably once you get that going, something else improves in the liver induced by FXR agonists that explains the higher level of ALT reduction. And this is reassuring because it tells you that beyond the defattening effect of these particular FXRs, you also have probably a specific anti-inflammatory effect. This is at least how I read it. And then moving on to the main problem here, very interesting. All these FXR second generation agonists actually seem to have pretty much solved the problem of the lipid changes, because if there is some LDL increase, it's really minimal and it's mostly restricted to those people that are not on statins. However, the one side effect that these second generation FXR still have to face is pruritus. And clearly there was a dose-related increase in pruritus, with almost 50% of patients that develop pruritus of different degrees, they're not all severe, in the high 2.5 milligram arm. So that tells you that this pruritus is still a class effect And it's the same with the drug that was presented earlier, which was nidofexor, the first day of the meeting. It's the same with what is it called, silofexor, and it is the same with tropifexor. Second generation FXR still have an issue with pruritus. So I guess the challenge now will be to find for each one of these compounds the right dose that reduces pruritus to an acceptable level and prevalence while still maintaining histological efficacy.
Hey, Vlad, just a quick comment on the last part with the AE profile. And I talk about this often, the juice worth the squeeze? And we know with FXRs, the higher you dose, the higher the AE profile. But I still think there's a home for these, particularly these second generation FXRs, because of what you just described in the ALT reduction, the PDFF reduction. And what are your thoughts on low dose combination therapy with something like Enanta's FXR?
You know, you got to be pragmatic. FXRs are the only drugs that have shown efficacy histologically in well-conducted large trials. So that's the starting point of all discussion. Now, the question is, do the second generation FXR have the same efficacy of OCA? So I think there are still useful drugs, even though you have all the others, paramimetics and so on. First, because they are the only ones that have shown efficacy. And second, because they have so many other effects, pleiotropic effects. Remember the improvement in intestinal barrier, improvement in fibrosis, in inflammation, a lot of other things that are beneficial. And I would ask the opposite question. I think all the other drugs need to raise up to the bar that was placed by OCA.
Yeah, no doubt. Based on what we know today, OCA's crossed the finish line first on fibrosis improvement. I think we'd be naive to think others aren't going to hit that bar though. So I'm thinking more down the road about how to mitigate the AE profile, but yet still get the benefit of what the FXRs have to offer.
Possibly, okay. But see, other drugs that induce massive weight loss did not yet hit that bar. So it might not be that straightforward. We'll see.
And we will. So with that, we're going to wrap up this portion of today's session. I'm going to invite Peter, Vlad, Don to step away for a little while. Stephen and Louise and I are going to go chat with our sponsor for a bit, and then we will come back for our final question. So now, having left our main discussion for the day, and we will come back to everybody for a final question, we are now moved on to our sponsored section. Our sponsors today are Sonic Insights Medical Corporation. They are the developers, manufacturers, and marketers of Velacor. About Sonic recognizing the rise of chronic liver disease, Sonic Insights is on a mission to reduce the disease by enabling routine assessment and management of liver health. Its breakthrough point of care product, the ultrasound solution Velacor, assesses and manages chronic liver disease with diagnostic accuracy that is comparable to MRI, the current non-invasive gold standard. And to the audience, if you think about the last conversation where we talked about the challenges and benefits of wide-fit scanning, this is a product that's created to work with that kind of a set of issues. Velacor is the first handheld 3D liver health assessment tool that is fast, accurate, intelligent, and portable. It provides a clear picture of the liver in real time so that healthcare practitioners can be confident in their diagnosis, treatment, and care. You can learn more by going to sonicinsights.com. And we have with us today George Aleftiris, President and CEO of Sonic Medical. Good morning, George. Good afternoon. Good whatever time of day it is where you are.
Hello, Roger.
And Dr. Michael Curry from Beth Israel in Boston, who is the primary author on a poster that Sonic is presenting this week at the digital ILC conference. Good afternoon. Good morning, Michael.
Good afternoon.
Okay. And Stephen and Louise have come over with us from the podcast to join in part of the questioning. So what we're going to do now is pretty simple. George and Michael will each take a minute or two to tell you about themselves and their backgrounds and why they're here and excited about what they're doing. And then they'll take about 10 minutes. George will talk a little bit about the company. Michael will talk some about the poster. Maybe George will talk about the poster. They'll do that however they wish. But the next 10 minutes belongs to you two gents. So go ahead. Have fun.
Well, thanks, Roger. And thank you for the opportunity to be here. It's an honor and a pleasure to participate in our first ILC conference that we have here. Not only a poster, but an exhibition as well. I'm the founding CEO of the company. I'm a serial entrepreneur. This is my third startup in the health tech space. We're based in lovely Vancouver. Hopefully you all will have a chance to visit Vancouver in the near future when COVID subsides, of course. And we are formed with the purpose to really revolutionize how the assessment of many chronic diseases that we afflict human health. Using our disruptive elastography solution. And we take the principles of MRE, basically external excitation, but marry it to handheld ultrasound. So if you are familiar with the butterfly, which is what's happening in the point of care ultrasound space, we are doing similar in terms of the liver health disease segment, using the accessibility, portability of handheld ultrasound, but retaining the performance characteristics of MRE and also MRI PDFF for liver health. We are a company that has foundations in the University of British Columbia. That is the technology platform that we've licensed. And as Roger mentioned, our first product is VelaCur, which is a 3D tissue assessment tool. And ultimately, our vision and mission is to reduce and eliminate chronic liver disease. But how we do it, our vision differs than existing tools. We want to enable physicians, not only hepatology, but all physicians to have access to this powerful tool to assess and manage patients with this disease, and even those that are actually at risk of disease. So really important to address this health crisis. And our procedure takes 5 minutes to perform. It can be delegated if the physician chooses, and it can be integrated in different type of environments, from outpatient to in-market or clinics itself. I'm really excited to be here, and happy to pass on the baton to Michael.
Thanks, George. I'm Michael Curry. I'm Director of Hepatology at Beth Israel Deaconess Medical Center and Associate Professor at Harvard Medical School. And I have a group of hepatologists that I oversee, about 13 hepatologists in Boston, Massachusetts, where we have a very large clinical practice of patients with all forms of liver disease. We have a non-alcoholic fatty liver disease center headed up by one of my colleagues, and we have been involved in the development of non-invasive markers for liver fibrosis for many years. Our center was pivotal in getting the transient elastography technology of FibroScan approved in the United States, and we have used this as well as other methods of non-invasive liver fibrosis assessment widely, including open access programs where these studies can be performed by our primary care practices to try and determine which patients have significant fibrosis and need to be referred on to care. It's been our pleasure to work with George and his team to develop and validate VelaCur over the past number of months, past year or two actually. And this is a very useful technology in our patient population. Thank you.
So to George and to Michael, first of all, we're delighted and honored that you've chosen to sponsor our podcast today at Digital ILC meeting. And we're delighted to announce that Sonic Medical will be becoming our first corporate sponsor. And I think what we do next is turn the floor back to you, gents, to talk a little bit about the paper and the exhibition that you have here at this conference.
Great. I'd love to discuss the presentation that we have had by poster at ILC, and these are the initial results of a clinical trial using VelaCur, developed by Sonic Insights to help measure liver elasticity and attenuation. As you've heard already, the VelaCur system uses algorithms which are similar to MRI elastography in order to measure the shear wave length within the liver to calculate liver stiffness, which we all know is a surrogate marker for fibrosis. Similar to MRI elastography, the patient lies on an activation pad, which is placed under the patient's back, and steady state shear waves at a frequency of between 40 and 70 hertz are transmitted into the patient. A full range image then is able to, ultrasound image is able to guide the user to the correct location for measurement of liver stiffness. And through a sweeping motion of a probe placed on the patient's anterior abdominal wall or over the right rib cage, a volume of about 100 millimeters cubed, or roughly 50% of an average male liver, is measured to produce the average volumetric spatial elasticity of the liver. The multi-frequency approach provides an opportunity for averaging the liver stiffness to reduce measurement of uncertainty. The attenuation is also measured from the ultrasound data captured simultaneously at the same time as the elasticity measurement. This trial was an initial feasibility study to determine if the system can differentiate liver fibrosis in patients who are with NAFLD or hepatitis C from healthy volunteers. And it was performed at 3 centers in the United States and in Canada. All patients had either a prior diagnosis of hepatitis C or had non-alcoholic fatty liver disease and had to have liver fibrosis as assessed by FibroScan with a score of greater than 8 kilopascals or higher within the previous 12 months. Patients' body mass index was on average between 23 for volunteers and 30 kilograms per meter squared for patients, but the highest body mass index we had in the study was 41 kilograms per meter squared. The study aimed to show that Velacur could discriminate healthy volunteers from patients with liver fibrosis with a high degree of accuracy. Velacur was able to differentiate these 2 cohorts in terms of fibrosis with similar AUCs to FibroScan, with AUCs in both groups being greater than 0.9 for fibrosis as well as steatosis. When we used a threshold of 6 kilopascals for fibrosis or elasticity and a threshold of 223 dp per m for attenuation, the specificity and sensitivity of Velacur to differentiate the cohorts is above 0.88 with a high sensitivity and specificity for both measurements. As an exploratory objective of the study, a limited number of patients also underwent MRI elastography for comparison with Velacur. In these patients, the concordance coefficient was approximately 0.87 in comparison to FibroScan's 0.5. When comparing the MRI PDFF, the correlation coefficient was 0.9, demonstrating an excellent correlation between Velacur's attenuation parameter as well as that, as compared to FibroScan, which was 0.6, when we compared the fat fraction as measured by MRI PDFF. In terms of safety, there was no adverse events experienced by any patient using the Velacur system. So overall, our pilot study has really shown that this portable system, which can be used as a point of care test and can be used by a variety of users, it has results that are similar to MRI for assessment of both liver stiffness or liver elasticity, as well as steatosis in both healthy volunteers as well as in patients. And presently, further work is underway to compare Velacur's ability to discriminate between stages of fibrosis and steatosis. Thank you.
Thank you, Michael. Louise, Stephen, whichever one of you would like the first question. Come on, Louise, go ahead.
I think it's good. No, for getting as much diagnostics into the primary care or a better setting that allows patients access to, or anybody access to information about the liver. Can I just ask, I couldn't see in the poster, I love the dynamic fat fraction correlation there, that's great, but with the FibroScan, was there a different, it doesn't state what probes were used, and there would have been a different probe on the higher BMI patients, for example. Now we know that with CAP, with the XL probe, you get a difference in readings and you'll get a different attenuation level for that. So how did you balance for that within the results that are displayed?
Yeah, Great question. I don't have the breakdown of what the difference numbers of patients who were assayed through the M as well as the XL probe, but from what I do remember, most of our patients with FibroScan were studied with the M probe and very few with XL, but I don't have the breakdown of numbers.
Fantastic, but it looks encouraging, and it's a nice poster. Well done.
And I'll make one comment as well from the company perspective. We use a curvilinear array, standard off-the-shelf probe. So there's one probe, be it you're a large person, moderate person, or a small person. It's one probe, so one measurement. There's no difference in terms of outputs.
Fantastic. So it doesn't matter about the rib space, because even FibroScan every now and again struggles with the absolute. Asian would be the key one, because they have such narrow rib spaces.
I mean, you still have to have the view to the liver itself, but we just need a view to the liver. We can do other measurements as well, but we use the rib cage as being markers, or the fixture where we rock the probe back and forth. But certainly the results can be obtained through different means as well, but we use the rib cage to rock back and acquire signal.
Yeah, it looks nice, I have to say.
Stephen?
Yeah, hey, Michael, this is Stephen. So congratulations on the poster. Also, George, to you as well. And I'm familiar with the work you guys have put out working with Michelle and Nizam and others out of your group. And so kudos for this proof of concept data. Just thinking about it as you're talking, and as George had introduced, I think there's some positives here that will give this modality legs. And I want to mention those before I ask a couple questions. So that is, it's portable. And I've had a chance to see this up front and up close, and it is small enough to carry around and use in clinic. And for that reason, I think it has a huge potential benefit to impact this disease. One of the things we talked about this morning, or that was mentioned at the ILC, and then we talked about in the podcast, was really the struggle we have at getting primary care to understand fatty liver as a real disease. And the paradigm is not such that you wait till the ALT is 200, and the patient is presenting with a swollen abdomen and sarcopenia and jaundice. Ultimately, it's trying to get at these people while they're asymptomatic, and they may or may not have elevated liver enzymes. And I think where we're able to give them a tool that's quick, it's readily available, it's easy to use, you don't have to have a medical degree to do it, is going to be really, really key in advancing. I also like the fact that you get two outputs. You get a stiffness and you get a liver fat content measurement through one probe. And it looks to me, focusing on the PDFF, that there is a strong correlation with that, not just in saying, yes, there is fatty liver, but maybe even potentially quantifying that degree of fatty liver. Rohit Lumba is a very good friend of mine and a pioneer in this field. And one of his comments he made this morning in the Q&A session was a comment that the higher the fat content, actually probably the more progressive the disease. And there may be something to that. We just have to look at it closer. I do think this is a proof of concept trial. And therefore, I want to ask you, Michael, some specific questions relative to where you see the next steps being made in relation to improving the clarity around the accuracy and the precision of your test. Because as you know, while liver biopsy is an imperfect gold standard, it is still required. This is a surrogate test, and it's going to need to be compared to biopsy, even if you use MRE, which I think is a very elegant way to have a proof of concept. So what are your plans? And the other question I have is diversity of patients. Obviously, we need to study patients with no disease, those with advanced fibrosis, and everything in between. And one of the things we're discovering with the CAP function on FibroScan, for instance, is that once you are cirrhotic, there probably is a false positive to that CAP. We have the benefit of doing MRI PDFF following CAP in our cirrhotic trials, and a lot of the CAPs are coming back over 300 with PDFFs less than 5%. So just your thoughts on those aspects, please.
Stephen, thanks for those comments. And as you said, this is a proof of concept study. And ideally, the study would be best done comparing this technology to liver biopsy. As you well know, it is very difficult to get patients to agree to liver biopsy, particularly with this disease state, and the frequency of liver biopsy, at least in our population, is quite low. We have patients who have undergone liver biopsies for diagnostic and clinical reasons. We have patients who are undergoing liver biopsies to enroll in clinical trials, and so we have some historic liver biopsy data that we have not fully analyzed yet. We don't have as much historic liver biopsy data on patients who've undergone a VeloCure assessment as we would like, but our plan is to continue to try and gather that information. I'll let George speak to this question, but we do not have plans at this present moment in time to do a VeloCure versus liver biopsy study. But we are interested in capturing any available liver biopsy study within a 6-month period that is available to us in patients who are undergoing VeloCure assessment. I agree with you as well that I think that as patients' liver disease progresses, there's some concerns with PDFF assessment. And our plans next are, we are moving forward to increase the number of patients who are having an MRI elastography as well as MRI PDFF collected at the same time as a VeloCure or within a short window of the VeloCure assessment.
And thanks, Michael. And I just elaborate a bit further. So we do have active site activation in our next study, which is comparing it to MRI PDFF. Now, biopsy is historical and it is being included, but when available. But the primary objective of the study is to compare to MRE, and the secondary is for comparison to MRI PDFF. Biopsy is still remains as an exploratory tertiary when available. Now, we do recognize that biopsy is important. So we have another program, as we call it, the early access program, that we're working with centers of clinical excellence and trying to increase our biopsy data set to complement what we do with the company-sponsored study, which is multi-site, and Beth Israel being one of the sites that we are working with Michael and his team. And that's what our current plans are. And that will also look at the patient diversity. It's important, certainly, that we recognize that although we may have 100 plus patients that were assessed in this study that's a poster, we do need more diversity in patient type subsets with different types of disease, for sure. And we're addressing that.
And I think, you guys have a plan that'll come. This is proof of concept. It's exciting to be able to show that you can detect both of these things, the stiffness and the fat. Because again, going back to primary care, I as an advocate in the NASH field need something that I can carry with me into PCPs and show them, guys, this is #real talk. It's not that difficult. Okay. Have your MA, have somebody do this before you see the patient. You have the results on your clipboard when you walk in the room, and you can make appropriate assessments relative to the findings you see from this handheld device. So I'm excited about it. I think it does have potential. There is a bit of development that needs to happen before that, as we talked about.
Yeah, and that's very important to have that sort of further validation to be able to go to the PCPs, for sure. And that's something that as part of our roadmap as part of market entry.
So George and Michael, because you weren't part of the previous section of the podcast, you missed an exchange between Louise and between Vlad Ratziu on this specific subject, where Vlad made the comment that FibroScan is an extremely technical tool best left in the hands of the specialist. And Louise agreed that it was a technical tool, but with training, it might not need a specialist to manage it. Have you started looking at what you think the requirements will be to get people in a primary care office adequately trained to use this tool appropriately?
So going back to what's happening in the ultrasound space, imaging interpretation it requires a different approach, because ultimately to train a user, you have to address the acquisition and interpretation. So we are integrating AI machine learning as part of a organ identification. So we can do segmentation of image and tell you have target liver, or you have target spleen, target different type of organs within the mix. Obviously with a PCP, you need to tell them what's liver and what's not. So the type of acquisition and target localization is shown. And depending on what the user is, an expert user may want to know, how much liver do I have in my measurement itself? Very important, but only for the expert user, perhaps. So we are addressing the acquisition from a software, but also from an interpretation, which I mentioned about that we're making those interpretation of image in real time. So it is within part of the appointment as 5 minutes, and make it really simple and delegable, and even that we're looking of how we can change how we deliver the solution. So moving away from a CapEx model to a service model. So there's no large upfront fee. It's actually a low upfront fee or no fee, ultimately.
Can I ask just a question on its operation? Obviously, FibroScan sends pulses through when you trigger the pulse. How is the pulse generated automatically when it hits the skin and you place the skin on the probe, or is there a sort of fire mechanism within that?
So similar to what's being done with MRE, is you use external excitation. Mm-hmm. So what Michael mentioned about the activation pad, that actually creates a shear waves through the whole abdomen. So You're not limited to any depth itself, because the imaging transducer, which we use, goes up to 15 cm. So it's applicable for all body types. So you really, unlike FibroScan transit elastography, which sends pulses, we do it through the external excitation approach.
Fantastic, because there are lots of things that I like about it, and it's highly positive. You can certainly work on brief interventional effects because you can see the results, and I think that's one of the strengths in my perspective for FibroScan, because you can give that feedback to the patient at the time, and they love to see something, and it's about the opportunity at the time. And I do hear Vlad in my ear talking from him, listening to him yesterday, talking about biopsy. In fact, he nearly did well to convince me, not that I would have a biopsy, but he had me going and was encouraging on it. So I understood his argument. And as I discussed with him earlier, that I do believe that FibroScan is still kept within a specialist technician doing it. So it's not that you just train anybody to do it. And I think Stephen may agree or disagree with me. Some of the worst people to do FibroScans are occasionally physicians, because they don't do enough of them. And I think we know that it's about the amount that you do for FibroScan. And then there's been some excellent studies through EASL so far, where the success rates in even morbidly obese has been well into the 90s, which is not what you see in clinical trials. And that may be just due to those sort of things. But Stephen, I just wondered, I think it was Nazim Nuruddin last night was talking about the changes to clinical trial endpoints, moving away from liver biopsy as those trial endpoints and moving more towards dynamic fat fraction. So the strength of this device appears to be its significant correlation with dynamic fat fraction. Do you think there would be the potential? I know Vlad would argue the opposite, but do you think there's the potential therefore to use that as this device as a surrogate of a surrogate?
I do think that's a great point, Louise. You know, the struggle we've had with the regulatory authorities is they're data-driven people, and they want to be very cautious in how they go about approving medications, and that's no different in NASH. The problem is we're faced with, as you noted, a surrogate of an outcome, and right now we don't have enough data to substantiate that. However, as we talked about on the podcast today with PDFF, and it's headed in that direction, I believe. And if it continues, you know, you guys are speaking of Sonic Insights, Michael and George. You guys are able to take Velacur and continue to generate data relative to what we see histopathologically across a broad spectrum of patients. You don't run into the false positive issue that potentially CAP has in cirrhotics, although that's yet to be proven. It's certainly anecdotal. And you're able to have a strong, you know, AUROC as well as sensitivity and specificity. I think that's going to be where you want to target this thing. The concern I've got with every one of these tests, and I've talked about it over and over again, is the positive predictive value when used alone isn't always where we want it to be. And I think that's probably not going to be any different with this one. The negative predictive value will turn out to be better than the positive predictive value. If I'm wrong, kudos to you guys, because you've now developed the first test that has a better PPV than NPV. But generally, that's not the way these things go. So it's long-winded answer to your question, Louise. I think it is possible, but got a lot of work to do with it first.
Well, let me ask George and Michael one more question, and then we should start to wrap this up, or else we're going to wind up with a 3-hour episode today. And that's not what these gents want or our listeners. So, George, apropos of all this, and Michael, how do you envision this moving forward, and how do you envision it being used in practice? You know, we talked about a few things. Who would be using it? What would they be testing for? How broad would be the target patient population? What do you envision?
Well, maybe I could speak just from the clinical perspective. Having used both FibroScan and Velacur personally and within our group, we have had better uptake, easier utility of Velacur than FibroScan. Right now, my clinical research assistant does all the Velacur studies, but without any difficulty whatsoever, and with very little training. So I do think that from a training perspective, and this is just anecdotal, that this is an easier machine to use than FibroScan. And as you quite rightly discussed earlier on amongst the group, is that FibroScan is technically more challenging, and we have our FibroScan technicians do all of our FibroScan studies, including our our open access program, for which primary carers refer their patients for FibroScans. Any of the machines that we send out into the community we have to send a technician out with the machine.
Yeah, and just to add to what Michael just said, I mean, ultimately, we do recognize that we do need to prove and win in the space of hepatology, but the long-term objective here is to roll it out into the other care providers that support hepatology, GI, endocrinology, and PCPs. Ultimately, if we're going to address the problem, we got to go to the source, and the source who holds those patients that are at risk or have disease, it's PCP and beyond. And that's what our objective is, to make both the quality, fidelity of information high, but ease of use really important and access as third.
So that sounds to me like a pretty good note on which to close, unless anybody start with Steve and Louise, and then to Michael, George, have anything else we feel the need to add? I'm going to take silence as assent. George and Michael, thanks for coming aboard today. Sonic, thank you for sponsoring this podcast and for becoming our partner as we go forward and try to educate the community. And we wish you nothing but success and good stuff as you move forward.
Well, thank you so much for the invitation, and look forward to our next discussion.
Excellent. Thank you, sir.
Thank you for letting us present.
Okay.
Thank you. Lovely.
Louise and Stephen, thank you for sticking around. Okay, that's a
pleasure.
wrap of the official part. Louise and Stephen, Michael, George, see you guys later. Louise and Stephen, could you stick around for just one sec? One more question for you. Thanks.
Nice to meet you guys.
Pleasure. Take care.
We lost Stephen. All right. Take care, George. Okay. We lost Louise too. Okay, Tom. We can put the other thing that I want to tape on here. It's just going to take me a minute to get organized to do it. Okay, so give me 1 minute and I will come back to you. Okay, thank you, everybody. We're back for our final question. I just want to go around and I want to ask, cross between the same question we usually ask and the question we started with, which is, is there one data point you saw today that is clarifying for you the kind of meta message of what's coming out of this meeting from where you sit? One thing you said, yeah, that makes this make sense. Or gosh, that makes nothing make sense. Either way. Brave one, go first.
So this is Donna. I'll start. I've had something on my mind. I feel that the Global Liver Institute's work in NASH, and certainly the way we constructed our NASH Council as a interdisciplinary, multi-specialty framework for the things that needed to happen in the field was the right thing to do, and has been a catalyst for a number of initiatives. When I heard Ezel ILF's presentation of their NASH agenda, it sounded very familiar. So I'm very glad that we could set out something that the other organizations around the world felt would be a good model to follow on as we build this field. So I feel really validated by the foresight and the approach that my team has taken at the Global Liver Institute.
I'll go next. And I think, not moving too far away from that, but I'm just going to go back to Dr. Rachel Pryke, who I commented on at the very beginning. And what she was very keen on and fighting back for GPs is they need better coding. It comes down to coding for the GPs as to how they can better manage it, building bridges with secondary care, education. But what she was very keen on was multi-morbidity management. So this multidisciplinary, multi-comorbidity management at a primary care level, she felt was very good. And that struck a chord with a lot of what we say on here.
Okay, that's great. Peter, Vlad, Stephen.
Well, I'm going to be a bit more obtuse and philosophical in regard to this question. And I'm going to quote none other than the Bible and say that I think that we are currently seeing through a glass darkly. I don't see everything falling together with clarity in NASH. I think we are moving through the dark and seeing some light, but all the pieces do not fall together for me at this point. We've made progress, but we shouldn't, in my view, we shouldn't kid ourselves of how much progress we've made, because I think we still have a long way to go. Dun, dun, dun.
I'll go next. And final say, which I think he'll appreciate. So I think, Peter, you're right. I think we're still searching for the end. I think we don't have a clear picture of where we are, but I think this meeting, what strikes me so far compared to AASLD and even EASL last year is there's more positive data. It's almost like we've figured out a little bit of the piece on therapeutics. We figured out a little bit of the piece on non-invasives. We're understanding that we're not reaching primary care the way we should, and we're beginning to figure that out. Do we have all the answers? No, but I do think there's a glimmer, there's a light. It's not bright and shining, guiding us out of where we are, but it's clearly moving in the right direction. And if I can go back to the Bible and add on to your comment, we have Moses has pulled us out of the wilderness, and we're headed in the right direction. I'll just say that.
Beware the golden calves, Stephen. But go ahead, Vlad.
Well, I don't know, Stephen. I hope it's not going to take 40 years to reach the promised land, because it'll be a little bit too long for us. And I, just like Peter, I sort of watched last week's and this week political convention, so I would be tempted to make the same comment about light and darkness, but I won't. I think the one thing that struck me in this meeting so far is one abstract, one presentation that showed something that I was saying all along and nobody wanted to hear, which is that people with NASH, which is a liver disease, die of liver disease. And this is a very nice study that was done, of course, by Vlad Ratziu, which has some sort of monopoly of these type of studies on large data sets and mortality rates in the population and cause of death in large national registries. And they have shown using 2018 data, not 1974 data, that patients with NASH die mostly of liver disease. 40 or something % of cases of death were related to the liver. And much less of that was related to cardiovascular or neoplasm. So that was extremely satisfying for me. And that is also germane to the comment we made about recognizing the metabolic factors and diabetes in that, but also recognizing that this is a liver disease, needs specific liver-directed therapies.
So, Vlad, I couldn't agree with you more, because that was the one I started with yesterday, where I said that really changed how I think people look at things. So I totally agree. The one I heard today actually is the one more about how much more primary care could do, assuming that they knew what to do after that, assuming that we're confident about what to do after that. And,
well, tomorrow the nurses will tell them what to do.
And I was going to say that sets up tomorrow very, very well. So thank you. And with that, thanks everybody again for a great day. We will be back here tomorrow, same time, same place. So enjoy. We will not be with Vlad, unfortunately. We'll be with Jorn Schattenberg instead, which is fortunate. And we look forward to that very much. So until then, everybody stay safe, surf on. Bye-bye. See you on the podcast.